Blood. What is blood? Well, it's a kind of connective tissue, and essentially it has four major components to it. So let's just put this down as the components of blood. Basically, when they take your blood and they do blood tests with it, one of the things they can do is they can take your blood and put it in a centrifuge. And what a centrifuge is it's a machine that swirls around. So some of you may use a centrifuge at some point in your careers if you go into any kind of research, or if you work in a hospital or healthcare or anything like that. So in the centrifuge, there's basically a test tube like this, right? And they fill it full of blood. And the test tube, you can put several of them in the centrifuge, and they're sort of held in there by a harness that allows it to swing. So when the machine that this is in begins to spin around, it's kind of like the graviton at the fair, and you get pushed out to the wall because you're stuck from the circular motion. So what happens is when it starts to spin like this, the test tube swings out and it takes on this shape. And it's sort of held in place in this harness. And so what's happening is there's a tendency for the heavier things in the blood to be pushed towards the outside, just like you're pushed against the wall of the graviton at the fair. So you get this hard, solid material building up in the bottom, right? And then you get this yellowy, straw-colored liquid left over in the top. And that liquid is called the blood plasma. So it's kind of like the fluid that blood is dissolved in, the blood plasma. And it's about 55% or so, according to this particular textbook I'm looking at. 55% of the blood is plasma. So my picture would be more accurate if I did something like that, right? About 55%. Okay, and plasma is mostly water. Essentially, it's water, and it might have things like dissolved sugars and salts and other things that might be in the blood, but it's pretty much water, about 80 or 90%. Now, the stuff that's left over, the red stuff, is sort of divided into three different types of cells that we notice in here. There are red blood cells, which are sometimes called erythrocytes, but we won't worry about that. Red blood cells, erythrocytes. And, well, I guess we can write it down. Erythrocytes. That's the fancy word for it. You're going to be a doctor. That's the word you want to learn. Red blood cells. What do they look like? Well, we can kind of draw one, sort of. But it's easier to look at them in actual pictures, so we'll do both. They're basically sort of flat disc-shaped cells, but let me draw. They have a sort of a dent in the middle. They kind of have this dent. So when you look at them from the top, right, there's this sort of region in the center of it's kind of depressed or dented. And actually the dent is on both sides, so I should draw my picture here with this little dent like this. Crayon, maybe I can shade it a little bit and try to get, so this is more like a dent in the cell. It's not really anything, right? So kind of like a darker region, I guess you would say. So they look something like that. And of course, the red blood cell is a kind of cell, but it's a highly specialized cell. Remember we talked about how cells choose a job to do, right? They become specialized. Red blood cells, they're manufactured in the bone marrow. So they're made by bone marrow cells, essentially. And they are specialized to the point where they don't even have a nucleus. So these cells are completely incapable of living on their own. without a nucleus, that means they have no DNA, no instructions. So they're made by the bone marrow, and they only last a few days at most, and then they die. Because they're so specialized in what they do, they have virtually none of the other functions that most cells would have. But they do have one important function, and that is that they contain something called, whoops, let's get my pen back, something called hemoglobin. Hemoglobin is a very complicated protein molecule. And in the middle of the protein molecule is an iron atom. So when you talk about iron in your blood, this is why you need it. To draw hemoglobin would be very complicated, but you could kind of think of it as one of those twisty-turny protein molecules like this, right? And then there's another twisty-turny protein molecule sort of over here, and if I'm not mistaken, I think there are 4 different proteins associated with this hemoglobin complex. And then in the middle, there's a little iron atom. There's an iron atom. And maybe it's 4 proteins or 2 proteins. To be honest, I'm not exactly sure if I remember, but all I know is it's a complex, I do know for sure it's a complex protein molecules that are interacting. It might be 2 different proteins, I'm thinking. But anyway, nonetheless, in the middle, there is this iron atom. And the iron atom gives the protein the unique property of being able to grab oxygen. This protein has a spot where oxygen can bind to it. But the bond is relatively weak. So the oxygen can bind to it, but can also be easily released. So it's a great molecule for picking up oxygen in a lung, bringing it to a cell, and releasing it so it can be then used by the cell. And of course, it can also bind carbon dioxide and release it. So this molecule in the red blood cells is basically what gives our blood the ability to transport carbon dioxide and oxygen. Oxygen to our cells and CO2 away from them to get rid of, right? As kind of a waste product. The waste product CO2 comes from the burning of glucose. Remember how we power our cells? We have glucose, C6H12O6, and we combine that with oxygen, and the products are carbon dioxide and water, right? So, plus energy. So we need this oxygen to go into the cells to make the energy, and then we have to get rid of all the CO2 that comes out, right? That's basically what's going on. That happens primarily inside the mitochondria of our cells. Okay, so that's a red blood cell, and I have a picture of one here. We can go over here. Red blood cells are right here. Here's a bunch of different pictures of them. These are drawings, but if I open up my ink here, no, I have to go back. Hang on a second. I have to turn on my pointer so I can move stuff around. Okay, there we go. I'm looking for an actual microscopic view. Here's a microscopic view, this one, right here. And you can actually see in this view little sticky bumps on top of the red blood cells. Those sticky bumps, remember how we said a cell membrane has those glycoproteins and glycolipids that stick out and form identifiers and sites for things to attach? Well, that's what you're seeing here. Some of those little molecules, these are called antigens, and these are what determines whether you have type AB, or O blood, and whether it's Rh positive or negative, and so on. These little things stick onto the blood, and of course different blood types have different antigens. We'll get to that part later. Right now, you can see how it has the dent in it. In this picture, it looks like the dent is really pretty much only on one side, because there are some that are not dented on the other side. If I go back to my pictures, or let's see what else is on there. Here you can see an interesting picture of the blood cells going through a blood vessel, and you can see the size. The cells themselves, there's only 3 or 4 wide, so this is probably a very small vessel. It might not be a capillary, it might be an arteriole or a venule, but it just gives you some perspective as to how big the actual blood vessels are compared to these cells, right? And that's basically how red blood cells work. Go back. And what we're going to do is we're going to draw some blood from a willing participant, and we're going to look at it under the microscope, and we're going to be able to see something that looks very much like this picture. This is actual blood through a regular light microscope. So we're going to do that. We're going to get somebody who's willing to have their finger picked, and we're going to draw a little blood. Well, it might have to be me, but we'll see how it goes. Anyway, that's red blood cells. So they are the oxygen-carrying transport cells, highly specialized, no nucleus. Look at them. You can't see a nucleus inside, right? They're just sort of an oxygen-carrying machine. Okay, so let's write that down. The hemoglobin, right? And maybe what we should do is we should add to that hemoglobin transports O2 and CO2 to and from. That's their primary purpose. All right. We also have something called white blood cells. White blood cells are a little more complicated to describe because they have a couple of different roles. And they have a couple of different names. The names of white blood cells are several different names. So let's just write white blood cells. Maybe you want to put a line under there. White blood cells. Yeah. The word we use to describe these primarily is leukocytes. So erythrocytes and leukocytes. But there are many different types of leukocytes that do slightly different jobs. But the main job that white blood cells do is they are part of our immune system. And their job is to identify and destroy foreign cells, or any kind of material for that matter. They will respond to anything that doesn't have the proper license plate. I'll explain that in a minute. In the bloodstream. Typically, the things that they would be attacking and destroying would be viruses or bacteria, but they will collect around just about anything. If you've ever got a sliver in your finger, then what happens, of course, is the sliver is covered in bacteria, because everything is covered in bacteria. It gets into your skin, and immediately your body begins to recognize that there's something here that doesn't belong. And of course, the way it recognizes that is by, remember, on the cell membrane, there are molecules, glycoproteins, glycolipids, that stick up and form different identifiers. So we all of our cells in our body have a specific arrangement of these that can be read like a license plate that says, you belong here. But a bacteria cell would not. So when the white blood cell is able to identify which cells are foreign, and it will then attack them. And typically the way it attacks them is by phagocytosis. So a foreign body will be, if it's small enough, a white blood cell will just kind of come around and engulf it like this, swallow it up, bring it inside the cell. It'll become a vacuole. It'll secrete digestive enzymes into there, and it'll break it down. But sometimes the object is bigger. And so what happens is the white blood cells will gather all around it in a big clump, and they will secrete digestive enzymes to break it down and destroy it. The white blood cells themselves often die in the process. And so what happens is in an infection, like when you get that sliver, all the bacteria are being attacked, and even the wood particles themselves could potentially be attacked by the white blood cells. And as the white blood cells are doing their job, they are dying on the spot too, and they're creating this nasty white substance that we call pus. Right? That's what that is. It's basically dead blood cells who've, given their lives to attack and destroy germs that are invading our bodies. A zit is a good example. The white goo in a zit is mostly dead white blood cells, and they're trying to get rid of whatever bacteria has caused the skin to become irritated in that region, right? That's basically what white blood cells do. So white blood cells are essentially a big part of the immune system. They're not really involved in transport. They're like policemen patrolling your blood and looking for, shady suspects who can be eliminated before they can cause any kind of sickness. Okay. Then, the last thing we find are something called platelets. Platelets are specialized little agents. They're not actually cells. They're cell fragments, but they serve an important role. These cell fragments, they're broken. They're sort of, they break off of cells within the bone marrow. Again, all of your blood is manufactured by the bone marrow, which is kind of interesting how your blood is made inside your bones, but that's how it works. Inside your bones, there are living bone tissues, right? Most of what we see in a bone is the outer part, which is the hardened calcium skeleton of the bone. But inside the bone, in the core, there are always living tissue cells that are producing the bone, but also making blood cells. These cell fragments break off, and so they're kind of weird little shapes, right? They're not like a typical cell, but they serve a very important role as they float around in the blood. If you have an artery that is cut, or a vein, whatever, and blood cells begin to leak out, right? Red blood cells. White blood cells will then also show up. The white blood cells, let me draw them in a different color. They'll show up and they'll start fighting bacteria that might be entering through the cut. But also the platelets. The platelets will flow through here, and they will begin to sort of stick together and clump up all the blood. And they'll create a clot. So they create what's called a blood clot. And the purpose of the blood clot is to quickly close the vessel and to stop the bleeding until the cells themselves can regenerate and heal the wound. So this idea of producing blood clots is what platelets are for. Platelets enable blood clotting. Now, there are some people who don't have the genes necessary to produce enough platelets. Remember, your genes are your instructions. And so in the genes, you have to have instructions that can sort of show cells how to make platelets. And some people don't have this instruction. So what that means is when they get cut, their blood does not clot very well. And so that means that they'll keep on bleeding. And it won't, like a normal person, sort of slow down and stop, put pressure on it, and then it goes away, and then it heals. These people are hemophiliacs, right? They have an issue with, they don't have the proper proteins in their blood to enable the clotting, right? Now, it might not be that they don't have the platelets. You have to understand that we're just learning the basics. And so lots of these processes are controlled and regulated by protein molecules, enzymes and things that can activate and turn on and turn off. So it could be that they're just missing some of the proteins. It's more likely they're missing a protein that enables the platelets to sort of do their thing. But nonetheless, they can't clot their blood. So that's kind of a problem. Blood clots, platelets. And that's essentially the four components of blood and basically what they do. So the red blood cells, the erythrocytes, are really the oxygen and carbon dioxide carrying part. Then we have these disease germ fighters, leukocytes, and then the platelets for clotting to enable wounds to be taken care of. The protein that helps the platelets clot is a protein called fibrin. So fibrin is a protein. This could be the protein that is lacking in people with hemophilia. Protein that enables platelets to form clots. Because the trick is, think about it. You've got platelets floating around. You don't want them just clotting everywhere. You only want them to clot at certain times when you have a wound. Otherwise, you'll have blood clots all over your body. So it enables platelets to clot. This is a protein that can sort of activate the clotting process. And that's essentially all we need to say about the parts of blood. But we will talk about blood types a little bit. So let's go down a little bit here and talk about what we mean by blood types. Well, you'll remember that on those red blood cells, we saw those little spiky dots, right? So red blood cells contain or are covered in, they don't really contain them. Let's put have molecules on their membranes. That form something called antigens. Antigens are essentially identifiers. They allow us, or our body, to identify these cells as belonging to us. And of course, there's different molecules, and different people have different antigens. And so, type A blood means you have a particular antigen that we have called, given it the name, the A antigen. Type B blood means you have the B antigen. It's just a different one. Some people have both. So they would have type AB blood. They have A and B antigens. Do I need to go back a bit? Sorry, I went too fast. Okay, where are you? Here? All right, I'll wait. A and B antigens, type AB blood. Some people don't have any one of these antigens. They have type O blood, which means they have neither antigen. So these are the antigens that determine your blood group. There's also another important antigen called the Rh factor. Rh factor. And those who have it, if you have this on your blood, then you are considered what they call positive, Rh positive. And if you don't have it, you are considered to be negative. So when you get your blood type, they're going to tell you that you're type AB, or O, or whatever, but they'll also say whether you're negative or positive. For instance, my blood type is O positive. So I don't have the A or B antigen, but I do have the positive Rh factor. Rh factor. Okay? Does anybody know their blood type? You can get it from your doctor if you were interested. We used to do blood typing back in the day, but I'm going to pause this. These antigens will determine how your blood cells behave. And if you don't match up the antigens properly when you're giving someone blood, the antigens can interact and cause the blood cells to stick together and become all clumpy and clotty. So if you had type A blood and somebody put type B blood into your system, let's say you were you had to have a transfusion or something, well, that blood is going to go and mix with your blood, and the different antigens are going to cause the blood cells to clot and stick together, which is not a good thing, right? It could be potentially lethal. So it's very important when we give people blood to match the type so that the antigens are not interfering with each other. Now, it turns out that type O is a little bit lucky because type O has neither antigen. So if you put type O blood in someone's body, there's no antigens in it that are going to interact. So we often call type O people universal blood donors. Universal donors. Because it doesn't matter. Type O is called the universal donor because type O blood can actually be given to anyone in a pinch. They still try to match where they can. But if you have type A and I give you type O, the O has no antigens on it, so there's nothing to interact and cause the problem. Now, people with type AB blood are also fairly lucky because they can get any blood. They're called the universal recipients because their bodies recognize the B antigen as theirs, and their bodies recognize the A antigen as theirs. So if you get either A or B, doesn't matter. Or if you get type O, doesn't matter. So if you have type AB blood, then they don't have to worry in an emergency which kind of blood they give you. They do, however, have to match up the positive and the negative for the Rh factor. So they'd like to match that if possible as well. So if you ever give blood, and if you happen to be type O, they're very happy when you give blood because it's a very useful blood for them to use. Right? Type AB blood is not very good to give other people because it has the antigens and it might cause a problem. But the type O is the best blood to get for a blood donor clinic because that blood can be used. Right? And they really appreciate type O blood. But of course, they also want the other types too, so that's important. And yeah, I think that's all we have to say about that and blood types and antigens. And we'll stop the video here.